Hello, students. Welcome to the class. Let us wait for 2 minutes. Let others be join. Then we'll start the class. Okay. Okay. Let us start the class. Okay. So before going to start the class, can you tell me whether I'm audible and screen is visible, students? Can anyone tell me whether I'm audible and screen is visible? Okay. Fine. Okay. Let us start the class. Okay. Fine. So let us start the class. In the last class, we have studied about the fungal diseases, right? So 1 minute. Yeah. We have started with the human health and diseases. In that thing, we have started the common diseases, right? Yes. So common diseases, in the common diseases, we have learned about the bacterial diseases, viral diseases, and the protozoan diseases and fungal diseases. These all the things we have learned, right? Okay. So here is the simple question for you all. Okay. So I think this is a very easy question. You can answer this question. The question is, which one of the following is a fungal disease? Okay. So many disease names, 4 disease name I have given here. In that, which one is the fungal disease we have to find? Okay, find out. So option A is tuberculosis. Option B is typhoid. Option C is chickenpox. And option D is ringworm. Okay. Can anyone answer me for this question? Okay. So I will run the poll. Meanwhile, you can answer me in the poll. Okay, fine. You can answer me in the poll and you can answer me in the chat box also. Okay, yeah. So which is the correct answer, student? Yeah, so many of them giving the correct answer. Yes, yes. So ringworm, right? Yes. So in the last class, we have studied about the fungal disease. Yes. So ringworm is a fungal disease, right? So when it comes to the tuberculosis is the bacterial disease, right? Bacterial disease. So that's why it is not a fungal disease. And typhoid is also bacterial disease. When it comes to the chickenpox is viral disease, right? Viral disease. Fine. Yes. The correct answer is ringworm. Okay. Fine. So let us move to the yes. So in the yesterday's class, this thing we have studied, but I have not shown this how it is going to be caused and the picture of that. So here you can see here. So this is a ringworm disease in the foot. Okay, in the fingers. And you can see here it is on the skin. Okay. So I have told you in the last class only that it is going to be caused by the microsporum, trichophyton, and epidermophyton, right? Yes. So in the last class also I have discussed that how it is going to be transmitted. So direct contact and use of clothes and towels of the patients, right? Okay. Okay. Fine. So these all the things we have studied this, right? So fine. So student, here you have to remember the causative agent. There are 3 causative agents here. Okay. Fine. Okay. So then today we are going to start about the immune system. Okay. So before going to start the immune system, here is the one more question for you. So largest defensive organ of the body is, can anyone think which is the largest defensive organ which is going to fight against the pathogen? Can anyone tell me? Yes. So see here, the correct answer is skin. Okay. Skin is the largest defensive organ of the body. Okay. Not liver, spleen, and intestine. Okay, student? Fine. So let us go to the next. So student, in this topic, we are going to study about in the immunity. Okay, mainly in the immunity. We have completed the common diseases, right? After that, immunity part will come. In that immunity part, we are going to complete immunity, types of immunity, innate immunity and adaptive immunity is there. So innate immunity, adaptive immunity, and also we are going to talk about the passive immunity, passive immunity and active immunity, active immunity. Okay, passive immunity and active immunity. So you are going to talk about allergy, how the allergic reaction is going to become. Okay, then autoimmunity, and also about the vaccination and vaccination. Okay, so these all the things we are going to cover in the immunity part. Okay, so passive and active immunity, allergy, autoimmunity, and vaccination. Fine. Okay, so let us talk about what is the immunity is. So we know that what are the pathogens, right? What are the pathogens? Pathogen may be bacteria, virus, or fungal, anything, okay, which is from the outside and which is causing the disease. We will call it as a pathogen, right? Disease causing agent, that we will call it as a pathogen, right? So actually, what is the immunity is? What is actually immunity is? See here. So the ability of the body to fight against the pathogen, okay? So that is called as the immunity. It was produced by the immune system, okay? So you see here. So our country was protected by our soldiers, right? Yes. So like that, our body is protected by our immune system. Immune system is act as a soldier to our body. Okay. So fine. Okay. So then what it is mainly going to be act as? Okay. Can anyone tell me what are the main functions of this immune system? What it is going to be act in our immune system? Act in our body. Can anyone tell me? Can anyone tell me? What happen if the immunity is not there means? What happen if the immunity is not there means? We are susceptible to the diseases, right? So if the immunity is there, we will not get the diseases. We will get the disease, but it is not going to be affect more, means much. Okay. So if the immunity is not there means we will suspect to the disease, right? Suspect to the disease. Suspect to the disease, right? Yes. So this is about the immunity. Okay, fine. Then coming to the how it is going to be act. Here it is going to be act in the three phases. Okay. So one we will call it as the recognition. Second, we will call it as the activation, and third one is the effector phase. Okay. So what happened? Some bacteria or the virus was entered into our body, right? Our body has to first recognize that whether it is a bacteria or fungi or it is related to our body or it is unrelated to our body. For example, if we take the food also, right? So, for example, that is a disease causing agent or the normal agent that has to be identified, right? So that is done by the recognition. Okay. So then second one is the activation. In the activation, the specific immune system is going to be activated. Either it is an antibody mediated or it is a cell mediated immunity. Okay. So the second thing is immunity is going to be activated. Okay. Then in the effector phase, what happening in the effector phase? The bacteria or the viral which is entered our body, the pathogen, it is going to be killed. Okay. Or it is going to be killed by our immune system. That we will call it the effector phase. What are the three phases, student? First one is the recognition, second one is the activation, third one is the effector. The bacteria any pathogen entered our body, so it was recognized by the immune system. Then our immune system is activated. Then our effector phase is the way the pathogen is going to be killed by our immune system. Okay, fine. Let us go to the what are the types of immunity? One is the innate immunity, and second one is the acquired immunity. So, what do you mean by innate immunity? Can anyone tell me what do you mean by innate immunity? Innate only is telling that it is from the by birth. Okay, innate immunity means, so it is the by birth. Okay, by birth only this immunity is going to become. For example, if you take our skin, right? Our skin, if you take the it is by birth only we are getting the skin, right? So then if you take the acidic pH in the stomach and the saliva in the mouth, and if you take the sweat, right? So this all the thing is going to be come from by birth only, right? And the urine if you take, and the vaginal secretion, right? This all the thing is coming from by birth, right? So that's why it is called as the innate immunity. And another thing we will call it as it is non-specific, right? Is that it is going to be specific? No. Okay. For example, our skin is acting as a barrier, right? If the enema pathogen come, for example, bacteria, for example, virus, if any fungus, it is going to act as a barrier only, right? It is not going to act as a differently for bacteria, differently for virus. No, right? Yes. So that is how our innate immunity or the non-specific immunity is going to work. Okay. It is not specific to the any of the pathogen. Okay. Fine. So other thing what you're going to learn. So that is about the innate immunity. So by birth only you're getting by till death. Okay. By birth to till death we are having this innate immunity. Okay. Fine. So then coming to what is the acquired immunity or the specific immunity. So acquired immunity is this immunity you are going to get after the birth. Okay, after the birth or in the life, what are the immunity you are getting, that we will call it the acquired immunity. You are acquired during your life. Okay, for example, you will get the bacterial disease, you will get the viral diseases, and in that the antibody is going to be secreted, right? So this all the thing, it comes in the acquired immunity. Okay, so the acquired immunity is going to be produce the immune cells which is specific to that type of the pathogen. For example, if you take in the acquired immunity, you will going to talk about acquired during the life. Okay, acquired during life or after birth. After birth. Okay, in that you can take the example of B cell and the T cell. Okay. For example, if you take the B cell means antibody production, right? The antibody are secreted in the acquired immunity. Okay. So not in the innate immunity. In the innate immunity, the antibody is not going to be secreted, right? So this is about and in the acquired immunity, there are two cells mainly going to perform the function. One is called as a B cell and another one is the T cell. Okay. The function performed by the B cell and production of the antibody, these all the things, we'll call it as a humoral immunity. Humoral immunity. 1 minute. Humoral immunity. Okay. And the performed by the T cell, we will call it as the cell-mediated immunity. Cell-mediated immunity. Okay. So this is what about the acquired immunity and the innate immunity. What I have told you about the innate immunity. Innate immunity is from the by birth only you are getting that, and it is non-specific. And coming to the acquired immunity, it is the acquired during the lifetime or after the birth. In that, there are two types of the immunity. One is the humoral immunity that is related to the antibody production, and another one is the cell-mediated immunity. So where the T cell is going to be working that. Okay. So example, you can take the cell-mediated immunity is you can graft rejection. These all the things will be performed by the cell-mediated immunity. Let us learn these all the things. Fine. So there are some examples and which are the barriers are there in the innate immunity. They are given that. Okay. So let us talk one by one. Okay. Yes. So see here in the innate immunity or the non-specific immunity, the first line of defense, it is also called as the first line of defense, and it is from the by birth only. Okay. So which are the barriers you are going to study here? One is called as the physical barrier. Okay. Second one is the physiological barrier. Third one is the cellular or phagocytic barrier. And fourth one is cytokinin barrier. And fifth one is inflammatory barrier. Okay. So these all the barriers which is present in the innate immune system. These four things, these five things are very, very important. Okay. They may ask you like this, which is the example for physical barrier? Which one of the following is example for the cytokinin barrier? These all the things. Okay. You have to remember these all the things. Okay. So let us study one by one. Okay. So can anyone tell me which is the correct answer for this? Which one of the falls above innate immunity? Can anyone tell me? Option A, inborn immunity. Option B is specific immunity. Option C is first line of defense. Option D is none of the above. Yeah, answer me fast. Which one is the correct answer? Can anyone tell me which one is the correct answer? Yeah, I will run the poll. You can answer me in the poll also, and you can answer me in the chat box also. Okay, fine. Yes, I'm getting the answers. Yes, very good. Yes. So the correct answer is, let us first, we will eliminate which is the correct answer. Okay. So yes. So when it comes to the innate immunity, right, it is the inborn immunity. That is correct answer, right? So first line of defense, that is also correct answer. And they are telling that in the option B, specific immunity. No, innate immunity is not a specific immunity, right? Only the acquired immunity is the specific immunity. No. So that's why option B is the correct answer, because they are asking here false, right? Not false, sorry, not true about the innate immunity they are asking. So that's why it is the specific immunity. Okay. So not none of the above also. Fine. So we will talk about the physical barriers. Okay. So which are the physical barriers? Okay. So whenever we are talking about the physical barriers, there are two main things. One is the skin. Okay. And second one is the mucus coating. Okay. So these both the things will act as the barrier for the microorganism, barrier for the pathogen. It will not allow any microorganism to enter into the body, and it will going to be replicated. So that's why it is act as the barriers to not allowing any microorganisms. Okay. For example, I have told you that which is the largest organ is, that is the skin, right? Fine. So see here, skin and mucus coating. Okay. Skin and the mucus coating, wherever you can see the mucus coating. So this is going to be act as the physical barriers. Okay. Fine. 1 minute. What is the main function of this? It will trap the microorganism. Okay. So it will trap the microorganism, and it will not allow any microorganism to enter our body, skin and the mucous membrane. Okay, fine. So let us go to the next. So that is the physiological barrier. Which are the physiological barrier? For example, the fever. Okay, fever. So what happens when you get the fever? Sorry, what happens whenever you get the bacterial infection? Your body temperature is going to be increases, right? Then only you will get the fever. So the fever is the indication of some pathogen was entered into your body. Okay. Yes. So okay, so that is the indication of something pathogen was entered to your body, right? Yes. So second thing was increase in the pH of the body, pH of the any secretions. Okay. So and you can take the pH of secretions of saliva and tears from the eyes and sebaceous gland. So these all the things is act as a physical barrier, physiological barrier. Okay. In the saliva, so there are the bacterial lysozymes are there in the saliva and in the tears. Okay. In the saliva and the tears, there are the lysozyme is there. Okay. So what is the main function of the lysozyme, student? Can anyone tell me? It will break bacterial cell wall, break bacterial cell wall. Okay. So the saliva, in the saliva, and if you take the tears, it is there is a main enzyme is present that we call as the lysozyme. Okay. So there, this enzymes it helps in the breakdown of the bacterial cell wall. So thereby decreasing the bacterial infection. Right. So tears and also the saliva. Okay. Then coming to the sebaceous gland. So sebaceous gland is going to secrete the sweat, right? Sweat. So our sweat is also having the high pH, and due to that, the microorganism cannot live on the skin. Okay. So that is also the one of the physiological barrier. Due to the high pH of the sweat, due to the high pH, the microorganism cannot grow. Microorganisms cannot grow. Cannot grow. Okay. So that is how our sweat gland is going to work. Okay. So sweating is good, right? Because whenever we sweat, it is going to secrete so whenever we sweat, the sweat is going to kill the microorganisms. Okay. So in the tears also, in saliva also, it is having the lysozyme, it is going to kill the bacteria. So fine. So I think you understood this. Okay. So let us go to the WBC. Okay. So why I'm telling this WBC here first is because we are going to talk about the cellular barriers. Okay. Before going to talk about the cellular barriers, first we will come to know what is the agranulocytes and the granular. Okay. Fine. So white blood cells or the leukocytes is divided into two. Okay. So one we will call it as the agranular, and second one we will call it as a granular. Okay. So here you can see that granulars is having the granules, right? It is having the granules-like structure in the cell, but granules do not have any granular structure. Okay. So when we are talking about the granulars, there are three types. Okay. One is a basophil, neutrophil, and eosinophil. Why it is called as a eosinophil, neutrophil, and basophil is eosinophil was observed after it is staining with the acidic pH, okay, acidic dye. Okay, so basophil was observed after it is staining with the basic dye. Okay, so and the neutrophil, it was in the neutral pH. Okay, neutral dye. Okay, so that's how it is got the name. Okay, eosinophil, neutrophil, and the basophil. Okay, students, here you have to remember in the leukocyte there are two types. One is the agranulocyte and another one is the granulocyte. When it comes to the granulocytes, which is having the granules-like structure in the cell, so those are examples of the basophil, neutrophil, and eosinophil. Eosinophil is stained by the acidic dye. Basophil is stained by the basic dye, and neutrophil is stained by the neutral pH dye. Okay, so then we will go to see here, the percentage is also given in the blood. Okay, in the blood, this all the thing is going to be present in the blood only. Okay, so leukocyte means white blood cells, right? So this all the thing is going to be present in the blood. Okay, so but if the percentages is differ. Okay, so when it comes to the okay, so welcome Janvi, Kudra, Kalyani. Okay, we have started the class and we are talking about the leukocytes. Okay, so leukocytes when we are talking about the blood, in the blood there will be red blood cells is also present, RBC and WBC is also present. Right. So WBC we will call it as a leukocyte. Okay. So in the WBC there are two types. One is a agranulocyte and another one is the granular cells. Okay, student. When it talking about the granulocyte, so one is a basophil, neutrophil, and eosinophil. Their percentage is also going to be changes in the blood. Okay, so the highest, whenever you are going to be infected, the highest number of the granular cells is neutrophils. Okay, in the blood test also you can see neutrophil percentage is highest. Okay, so neutrophil 60 to 70%, and eosinophil, student, eosinophil is going to be act only in the allergic conditions. Okay, allergic, and when you are infected by the platyhelminthes, okay, on the time eosinophil percentage is going to be increases. Rather than that, its percentage is 2 to 4% in the body, okay, in the blood. When it comes to the basophil is also in the allergic reaction. So it is about the 5 to 1%. Okay. Whenever you get the allergic reaction, the main cell involved in the allergic reaction is the basophil. Okay. In the basophil, it is consisting of these certain chemicals like histamine and serotonin. Okay. So these all the things is going to be released during the allergic reaction from the basophil cells. Okay. Basophil and the mast cells. Then coming to the agranular cells, in the agranular cells, there are two types. One is the lymphocyte and another one is the monocyte. So when we are talking about the lymphocyte, there are three types. One is a T cell, B cell, and third one is the natural killer cell. Okay. NK cells means nothing but natural killer cells. Okay. Natural killer cells. Okay. NK cells. These all the things will come under the lymphocyte. So then coming to the monocytes. Okay. So student, here you have to remember that this monocyte is a agranulocyte. Whenever it is in the blood. Okay. Whenever it is in the blood, we'll call it as a monocyte. Okay. When this monocyte entered into the tissue. Okay. So when this monocyte entered into the tissue, we are going to call it as macrophages. Okay. So we are going to call it as macrophages. Okay. So this is all about the leukocyte. What are the things you have to remember here? Agranular cells are lymphocyte and the monocyte. In the lymphocyte, B lymphocyte, T lymphocyte, and the natural killer cells. And in the granular cells, basophil, neutrophil, and the eosinophil. These all the things you have to remember. Did anyone having the confusions here? Can anyone tell me? Surya Dipti, Suhani, Shubhalakshmi, Swapna, Kalpita. Can you answer me in the chat box, student? Do you understood this? Do you understood this? Can anyone tell me? I need answer from everyone. Fine. So we'll go to the next then. Okay. Which are the mainly cellular barriers which is present in the blood? Okay. So student, these all are the cellular barriers which is present in our blood. Okay. So which are those? For example, you can see here they are given that cellular barriers, neutrophil, monocyte, macrophages, and the natural killer cells. Okay. So these are present in our body. So that is in the blood. Okay. These are present in the blood. Okay. So you can take monocyte, lymphocyte, neutrophil, eosinophil, basophil, macrophages, erythrocyte, and the platelets. So, but mainly the phagocytic barriers are neutrophil, monocyte, macrophages, and the natural killer cells. For example, if you see any bacteria was entered to our body, okay, why we are going to call it as the phagocytic cell? See here, so this is the blood, okay, this is the blood. Some bacteria entered into the blood, okay. Our body, our blood is having the soldiers, right? So that is the immune system we have. So these all are the soldiers only, neutrophil, monocyte, macrophages, and NK cell. They came and they saw this bacteria. Okay. So they came and they saw this bacteria. What they will do? Simply they will say no. Okay. So they will eat this bacteria. Okay. So how they are going to eat this bacteria? See here. They are going to engulf this bacteria by the process of phagocytosis. Okay. The process we will call it as phagocytosis. See here. You know, in the plasma membrane, we have studied about the invagination of the plasma membrane. So how it is going to be take the outer particles through the plasma membrane, right? So that is what we will call it as invagination. So after that, it is going to be taken inside the phagocytic cell. See here. Inside the phagocytic cell. 1 minute. See here now, the bacteria was entered into the phagocytic cell. So what will happen now? So in the blood only, this all the thing is going to be happen in the blood only. Okay. So now this bacteria, so this thing, it is covered by the phagosome. Okay. 1 minute. So this bacteria is now covered by the phagosome. Okay. Phagosome we will call it as. Okay. So there are in the cell, there are the lysosome is also present, right? Lysosome. So now what will happen? It is going to be fused with the lysosome, okay, which is that phagosome and lysosome is going to be fused. Okay, see here. Here now, there's bacteria. Then bacteria is having the lysosome, and the phagosome is going to be fused. Okay, student, remember what will happen in the when the phagosome is fused with the lysosome? It is having all the enzyme which will break down the bacteria, right? In the lysosome, there will be a protease enzyme, there will be a nuclease enzyme. So this all the thing, what it will do, it will break the bacteria into the small pieces. Okay, so it will break bacteria into the small pieces, right? So now bacteria has been dead now, right? See here, the bacteria is came into the pieces, right? So what we'll do now? So this phagocytic cell is going to be show this. 1 minute. Okay. So now what happened? This phagocytic cell is going to be taken this some of the fragments of the bacteria. Okay. So have you know about the this Angulimala thing, right? So whichever the persons are killed by the Angulimala, he will remove the finger and he will make it to the this garland, right? So same thing, this all the things is going to be do. Okay. So what it is going to do, what it is going to do, it is going to kill the bacterial cells, right? It will take some of the fragments of the bacterial cell and it is going to show into the on the surface. Okay, it is going to be show to the surface. Okay, it is going to be, see here, 1 minute. See here, it is going to be show on its surface. Okay, so this is how this phagocytic cells is going to be work. Okay, to whom it is going to be show? It is going to be show to the B cell or the T cells. Okay, so these are the bacteria which is going to be entered our body. So entered into the blood. So I have filled them. So this is the bacterial fragment. It is entered our body. It is going to show like that. Okay, some other bacteria same is there, right? Which is not killed by the phagocytosis, which is not killed by the phagocytic cell. It can be attacked by the B cells. Okay, fine. So this is what the work is going to do by the phagocytic cells. Okay, so that is the phagocytosis. It is phagocytosis or cell eating, we will tell, right? Phagocytosis or cell eating. Okay, student. So this is how the phagocytic cell is going to be work. Phagocytic cells are neutrophil. Neutrophil is a very abundant, and monocyte, when it entered into the blood, we are going to call it as macrophages, and macrophages in the tissue, right? Yes. So then NK cells. Okay. So NK cells, how it is going to kill, right? Okay, I will tell you. So this NK cells, this is the NK cells. This is going to produce some type of the chemicals. Okay, some type of the chemicals like perforin. Okay, some type of the perforin and granazymes. Okay, so due to the release of this perforin and granazyme, there in the bacteria, what will happen? It is going to create some types of the pores. Okay, see here, it is going to produce the some types of the pores in the bacterial cell. Then it is going to kill the bacteria. So what will happen if the cell was burst means, right? If the hole was happened to the bacterial cell means all the content will be removed from the bacterial cell and it is going to be died. Okay, so it was done by the natural killer cells. Okay, student. So this is about the cellular phagocytic barrier. Okay. So here you have to remember neutrophil, monocyte, macrophages, and NK cells. There are the another cells are there. So those are the dendritic cells. Dendritic cells. Do you understood this, student? Do you have any confusions here? Can anyone tell me, do you have any confusions here? Do you have any confusions here? Suhasini, Suhani, Suryadipti, Subhasmita, Swapna. Answer me, student. So these are the acting as the cellular barriers. Okay. These are the cells. Fine. Next, coming to the cytokinin barriers. What are these cytokinin barriers? Okay. So these cytokinin barriers are acting as a message. Okay. So in the cell, in the blood, we are having the we are having the white blood cells, right? We are having our immune cells. So how these cells is going to be communicated? Okay, these are going to be communicated through the cytokinins. Okay, cytokinins. So these are the chemicals which are released by the cells. Okay, these are the chemicals, or we will call it as a chemical messengers. Okay, chemical messengers. For example, in this, the interferon will come, and interferon and tumor necrosis factor. Can anyone tell me which are the things you know about the cytokinin barriers? Can anyone think and tell me which are the cytokinin barriers will come? I need answer from you people. Can anyone tell me what is the main function of the cytokinin barriers? If you know, can anyone tell me? Okay, fine. So see here, these cytokinins are the low molecular weight proteins. Okay, these are the low molecular weight proteins. Okay, student. These are going to be active. Okay, these are going to be helpful in the differentiation, and proliferation, and the proliferation of immune cells. Okay, of the immune cells. Okay, for example, any bacteria was entered into our body, some cell is going to be activated. For example, if you take the macrophages and the some other cells, neutrophil, these are going to be engulf the bacteria, right? Then it is going to activate the B cell, right? It is going to activate the B cell, then it is going to be differentiate and proliferate, right? For that, this cytokinin is going to be help, okay? This cytokinin is going to be help in the differentiation and proliferation of the B cell and also the T cells, okay? So in the cytokinin barriers, we can talk about the interleukins. Interleukins, okay, student? Interleukins is going to be act in the differentiation and proliferation. Okay, differentiation and proliferation is going to be done by the interleukin. Okay, sometimes what happens, our cell is going to be infected by the virus, right? So for example, see here, this is a cell which is infected by the virus. Okay, so now the virus infected cell will send the message for other cells in the body. Okay, so I have infected by the virus, so be careful, the virus will come and infect you also. Okay, it is sending the message, right? So that thing we will call it as that message we will call it as the interferon. Okay, see here, here I have given that interferon are the proteins secreted by virus infected cells. Okay, see here, interferon, this cells was infected by the virus, then it is sending the message to the these cells are not infected by the virus. Okay, it is sending the message to that. So the virus is there in the blood, so be careful, be ready for the attack. Okay, like that it will send the message. Okay, fine. So this is about the interferon. Interferon was released by only virus infected cell. And what is the tumor necrosis factor? The tumor, what is the one minute? Tumor necrosis factor. Okay, this is also the one of the type of the chemical which will kill the cells. Okay, so which will kill the cells. So that is what we will call it, the tumor necrosis factor. Okay, so which are the things we have studied in the cytokinin barrier, student? One is the interferon, and second one is the TNF, tumor necrosis factor, and the third one is the interleukin. Interleukin was helpful in the differentiation and proliferation of the immune cell. TNF in the tumor necrosis, which is killing the cell, and interferon was only for the virus infected cells. Okay, fine. So then here is a question for you. So can anyone tell me which one of the following is not a physiological barrier? Student, you have to remember that, which is the, what is the difference between physiological barrier and also the physical barrier? Physical barrier. Okay, fine. I will run the poll. You can answer me in the poll, which is the correct answer. Fine. Answer me in the poll. I'm running the poll. Answer me in the poll fast. Okay. So the correct answer is mucous membrane. Right, student? Yes. The correct answer is option C, mucous membrane. Why the option C is correct answer? See here. So I told you that whenever we are talking about the physiological barrier, physical barrier, there are only two things are there. Physical barrier, one is the skin and another one is the mucous membrane, right? 1 minute. Mucous membrane, right? So these two things is acting as a physical barrier. So then coming to the tears of the eye and HCl in the stomach, HCl in the stomach and the saliva. I have told you that tears and the eyes is consisting of the enzyme. So that is called as lysozyme, right? This lysozyme will kill the bacterial cell wall, right? So that's why this both the three things, tears of the eyes and saliva and the HCl. So these all the things will come under the physiological barrier. So that's why the correct answer is mucous membrane. Okay, fine. So then coming to the complement proteins. So student, so still that we are studied about the what are the physical barriers, what are the physiological barriers, and which are the cytokinin barriers. Now let us talk about complement protein. Okay, so these complement proteins are secreted by our liver. Okay, so when we are talking about the complement protein, so it will complement our immune system work. Okay, so that's why we are calling as the complement protein. It will increase the immune system to work against the defense. Okay, work in the defense. Okay, so there are 30 proteins, 30 plasma proteins, which are secreted by the liver. Okay, which are secreted by the liver. So which are inactive first. Okay, first they are inactive, and during the activate they will activate, then they will work. Okay, so they are inactive at the starting. Okay, so they are synthesized by livers. Okay, what is the main function of this complement protein? So this complement proteins, it will create the pores in the bacterial cell wall. And same thing, what happens if they create the pores in the bacterial cell membrane means, so the osmolarity is going to be differentiate. The equilibrium of the osmolarity is going to be decreases or increases. Thereby, the which are the liquids which is outside, it will come inside the bacterial cell. It is increases the volume in the bacterial cell. Then it is going to be burst the cell, or the things which is present in the bacterial cell, all the organelles and all the liquid things, it is going to be removed from the bacterial cell. Thereby, it is going to cause the plasmolysis. Okay, it is going to be kill the bacteria. Okay, so that is main function of the complement protein. So see here, it forms the transmembrane pore in the microbes and burst them. So this the bacteria means what it will do, it will simply create the pores in the bacterial cell. See here, it is going to create the pores in the bacterial cell, and it is going to be release the contents, release content in bacterial cell, release the content which is present in the bacterial cell. Thereby, it is going to cause the death of bacteria, death of pathogens, we can call this. Okay, death of pathogens. Okay, so this is how our complement protein is going to be work. Here you have to remember that 30 plasma protein which is secreted by the liver. So first they are inactive, then later it is going to be activate. Okay, fine. So let us talk about the inflammatory. What is the inflammatory? So do you remember, student, whatever the, if you're hurted by something, or if you any bacteria, or if you were bitten by the mosquito, suddenly the swelling is going to be happen, or suddenly the redness is going to be happen, right? So that is the indicates that the tissue was damaged. Okay, inflammation is nothing but it is the sign of tissue damage. Okay, it is sign of tissue damage. Okay, it is sign of tissue damage. Something happened to our skin, or some tissue was damaged here mean, so the sign was going to be given like pain, swelling, and the redness. So the all the things we are going to call it as an inflammation. Okay, see here, something was came here, right? Something is going to be hurted, or any bacterial was sucked the blood means on the time it is bacterial bite, some insect bite. Okay, insect bite. Okay, insect bite. So what will happen? The harmful things entered into the our blood. So through the insect saliva, it is going to be come into our blood. So thereby it is going to damage the tissue. Then it is going to then see here our immune cells, our white blood cells is going to be entered and going to the infected area. Okay, so thereby it is going to cause the swelling and the redness. Okay, so this is about the inflammation. Okay, inflammation is nothing but sign of the tissue damage. It may be a pain, swelling, and redness due to the vasodilation. Fine. Let us talk about the acquired immunity. What is the acquired immunity? I have told you that. So it is going to be acquired in the lifetime and it is specific in nature. And I have told you for the bacteria, if the bacteria was entered, it is going to be act differently for the bacteria, and for the virus, it is going to be act differently, and for the pathogens like platyhelminthes and for the fungus, it is going to act differently, right? So that's why we will call it as specific in nature. Okay. So then supplement the protection provided by the innate immunity. Okay. So it will supplement the protection supported by the innate immunity. Then coming to the acquired immunity, the main thing we have studied in the innate immunity is it does not have any memory. Okay. So it does not have any memory, but our acquired immune system having the memory. So that's how it is going to help, right? Whenever the person is affected by the corona first time, it is going to be severe. Means in the first time that if was affected by the corona means it's going to show the symptoms and all the things. So in the second time, it is the symptoms was not that much severe, right? So why? Our system, our immune system is having a memory. So it is going to be increase the immune system in the very less time, right? It is going to be increase our immune cell in the less period of the time, right? So that is how we will call it as the memory based. Okay. Then identify the self and non-self. Okay. In the acquired immunity, I have told you that so that I have told you that cell mediated immunity, right? Cell mediated immunity is there. So for example, if you see whenever the person was having the damaged kidney or damaged heart something, so the transplantation is going to be done, right? So whenever transplantation is done, that is from the outside the body, right? Our body will recognize it is from the outside the body, then it will kill that. It will damage that organ which is transplanted. Okay. So which is the immune system is going to work there? So that is the cell mediated immunity. So that is also the acquired immunity. Okay, fine. So that is why it is going to identify self and the non-self. Okay. So then it is works on antibody reaction. Okay. Antibody reaction. So that is humoral immunity I have told, right? So this is how it is going to be work. So acquired immunity, what you have learned here, it is the acquired during the lifetime. It is specific in nature and it is a memory based and it will recognize self and the non-self. Okay. Student, do you have any questions here? Do you understood what are the things we are learning here? Can anyone tell me? Jainvi Damantri, Sonali Biswal, Jainvi, Surya Dipti. Can anyone tell me whether you understanding or not? Fine. So let us talk about the humoral immunity. Okay. So what is the humoral immunity? Humoral immunity is mainly based on the antibodies. Okay. It is based on the antibody production. So student, in the humoral immune system, mainly the cell involved here is the B cell. Okay. So what happened in the humoral immune system? Let us talk about this. Okay. See here. In the humoral immune system, what happened? Any bacteria or something, virus are entered into our body. Okay. So now the cell was infected. Okay. The cell was infected. So that is you can take the macrophage or something. Okay. Macrophage or any cells or the normal cells. Okay. Normal cell will also show the antigen representing antigen fragments to the B cell. Okay. See here now. B cell, it was showing to the B cell. Then the cytokinin was released by the B cell. Now, activation of the B cell was happened. Okay. So where this activation is going to be happen mainly in the lymphoid organ. Okay. Lymphoid organ. Okay. So B cell was activated. So mainly the B cell is going to be activated by T helper cells. T helper cells. Okay. So T helper cell activated the B cell. So now B cell was activated into the 2 cell mainly. Okay. One we will call it as the plasma cell and another we will call it as the memory cell. Okay, one is plasma cell and another one is the memory cell. So memory cell is kept like that only. Okay, it will be like that only. So whenever the same bacteria or same pathogen was affected next time, the memory cell will remember that and it will synthesize, again it's converted into the plasma cell and it is secrete the antibody. Okay, so but see here, the plasma cell was secreted, right? It goes on secreting the antibody. Okay, so see here, you have to remember the plasma cell will secrete the antibody. Okay, so now what will happen? This antibody is going to be coat on the bacterial cell. See here, whenever the antibody is secreted, what happened? So the bacteria was entered, right? This bacteria was entered into the blood. So this bacteria was coated by the antibody. Okay, this was coated by the antibody. Then it will attacked by. Now this was attacked by macrophage cells. Macrophage cell. Okay, so it is going to be neutralize the antigen. Okay, so this antigen was entered. 1 minute. Antigen was entered into our body. Now our antibody is going to coat on that antigen. Then it was attracted to the macrophage. Now macrophages will come and it will eat that bacteria. Okay, so we have studied in the phagocytosis, right? So that is how the macrophages will eat this bacteria. Okay, students? Fine. So this is about the humoral immunity. In the humoral immunity, we are going to talk about the antibody structures. Okay, so can anyone tell me whether you understanding or I need an answer from you? Barsha, Janvi Dhanmantri.
Yes.
Can anyone tell me? Fine, student, fine. Okay. So let us talk about the antibody structure. Okay. So student, remember here, our antibody structure is made up of the two chains. Okay. So see here. So this red color I'm writing, right? So see here, this is a heavy chain. Okay. So this is the heavy chain. Okay. It is made up of the two heavy chain. Two heavy chain. Okay. Two heavy chain. And so this thing you are seeing, right? So this is the another one. 1 minute. I'll color by another color. Okay, wait. 1 minute. Green color I will take. Okay. So this is what we will call it as the light chain. Okay. So the antibody is having two heavy chain and two light chain. Okay, student. Two heavy chain and the two light chain. Okay. So see here. So the light chain and the heavy chain is having the main structures. Okay, so this region is there, right? So this is the region. So this region we will call it as the variable region. Variable region on all the types. Okay, this is the heavy chain, right? This is the heavy chain variable region. So this is the light chain variable region. Okay, so this is the light chain variable region. Okay, so this is also. So this outer part, okay, this outer part is there, right? So this is heavy chain variable region, and this is the light chain variable region. Okay, this is the light chain variable region. So other than that, okay, only these things, okay? Other than this thing, so the remaining things is there, right? Remaining things. So this is the thing. And remaining this, all the things, we will call it as the constant region. Constant region. Okay, constant region. So one is the variable region, which is outside, and this thing, both the things, all the things, we will call it as the constant region. Okay, so fine thing. Fine, student. We will learn this all the things in the coming class. Okay, so if you have any questions, you can ask me. Okay, so that's why, student, in the antibody structure, we'll call it as H2 and L2. Okay, H2 and L2. Okay. So this is the structure I have written, right? Wait, I will tell you. See here. 1 minute. Yes. See here. So this is the structure, right? This is the structure. This is the heavy chain. Okay. This all the thing what I have written is a heavy chain. And here I have written, right, red color. So this red color structure is the light chain. Okay. Fine. So let us talk about this all the thing in the coming class. Okay. Fine, student. This is the structure we will study in the next class. Do you have any question? You can ask me or else you can leave the class. Okay, wait 1 minute. Okay, so here where is the variable region, students? This is a black color, okay? Wherever the black color I am quoting here, this is the variable region, variable. Okay, this is a variable. So this is variable, okay? Other than this all the things are the constant region. Okay, these all are the constant region. Okay, so this is the light chain. Okay, so this is the heavy chain. Heavy chain. Okay, student? Fine. So these all the things we will study in the tomorrow in the next class. Okay. Thank you so much.